Have everybody. A warm welcome. Good afternoon to all of you. Most of you I've never seen in this room before. This is where we have been able to do lab meetings recently. By the way, can you hear me online? Yeah?
I hear you. Can you guys,
okay.
can you
Good.
see me? Just so I know, am I there in video? Because I don't have there's no video showing up for me here.
I cannot see you. I only see your name.
But you can see our room?
I see Steve. Yes, I see you guys in the room. I see the slides.
You can see me wave.
Yes.
Okay.
But my video is showing up blank.
Okay.
Just so you know. But I see everybody. Okay.
Maybe we just have to do with that then.
Sorry to interrupt. Go ahead.
Yeah. Okay. So this is Stephen Shuken's thesis defense. Very excited. Obviously, a very momentous event, and I'm really tremendously excited about this occasion. Stephen has been absolutely fantastic student and has achieved really great things. And I just want to give you a flavor and an introduction. And I would also like to thank the thesis committee members, and particularly Noah Burns, who sort of let Stephen go. As you may know, Stephen actually started out as a chemist. And one day he knocked on my door and said, I want to do what you do. And I said, okay, wait, not so fast. First, we have to train you. And I just show you a few examples here of the rigorous training that we go through with our students. So usually they start out as bus boys. And you see after a few weeks of training, I think Stephen mastered this pretty well. We have events where they have to learn to socialize. Very important aspect for doing research, especially in today's sort of team-driven research. They have to learn how to act surprised and excited when a team member presents their lab data. So this is one of these practice events here. Winning support, charming others, engaging, very important, of course. And then learning to introduce yourself properly. So I just put the microphone here. Can you hear this? Stephen, can you help me? So the students have to learn how to help their PIs?
Yeah. Yeah, that's good. Can we turn it up? Yeah.
I'm House Dark, Westeros. My name's Steven. I'm House Dark, Westeros. My name's Steven.
Yes, so you get the gist here.
Westeros.
And then rapidly changing costumes.
It's been over like 20 seconds.
Can you just relax for like... Thank you.
That doesn't even cover your beard.
So you also note that this is pre-COVID. He actually doesn't know how to put on a mask. And then also promote your ideas, very important. And here we actually recruited Scott Shuken, Stephen's twin brother. Is he here? We haven't met. Oh, okay. Hey, Scott. So here they just, they have this podcast where they just make up things and talk nonsense for hours, actually. And I invite you to listen to them. So that's a good practice, of course. And then here, this is boot camp. So this is actually our lab Olympics. And so the person says, Stephen, the samples, we're waiting. So Stephen has to put these samples into a rack in record time. And you can see he can do this pretty well after a while. And then finally doing stuff, right? And usually when I talk to Stephen and asked him, what are you doing? He said, oh, I'm fixing the mass spectrometer. So that's a bad joke, but Stephen has been wonderful at doing this. And then lastly, the mission abroad. So here I had to cover the identity of the other person. But here Stephen infiltrated the lab of Paola Picotti at the ETH in Zurich. And this is the first picture he sent me. I still don't know what he wanted to say with that, but it says "Fight Cops," a graffiti somewhere in Zurich. And then here the team that he infiltrated there. And lastly, he passed, he fully integrated into the lab and has become a really incredible team member, mastered many of sort of basic neurobiology techniques and learned to work with mice and all kinds of different experiments and creatures. And we're going to have a reception for him. And again, I'm really delighted and so glad that you joined the lab and look forward to your defense. Thank you.
Can everyone hear me? Great. Thanks so much, Tony. Oh, I'll use the HDMI one. Thank you. Oh, my trackpad isn't. Yeah, okay, now we're on. Okay, and people online can hear me?
Yes.
You got thumbs ups. Thank you so much, Tony, for that hilarious and kind introduction. I actually had a very unfair advantage coming to the lab because I was a real busboy at the Coho Cafe in Sammamish, Washington before joining the lab. So I can finally get that off my chest. I want to thank you all for being here as well. I'm very excited to share my research on brain aging, which I studied by looking at the cerebrospinal fluid using mass spectrometry. But first, I thought I would give a little bit of context and explain how I got here, why a chemistry student such as myself is giving a talk on such a topic out of field. My background is in synthetic organic chemistry. Organic chemistry is the chemistry of carbon-based molecules, which make up all living things. And synthetic organic chemistry is the study of synthesizing or building these molecules step by step. We do this by performing synthetic reactions, such as this one here, called an acetylation reaction. In this reaction, we're building a bond between the oxygen atom on this starting material on the left and a carbon atom in acetyl chloride. And this is actually a quite important reaction. The starting material on the left-hand side is salicylic acid, which is an anti-fever medicine used in the 1800s. But when people took it, there was a side effect, which is their throats would get irritated. Performing the acetylation reaction on this compound makes aspirin, otherwise known as acetylsalicylic acid, which has the same anti-fever properties, but no throat irritation. In undergrad, I studied natural product total synthesis, which is the subfield of synthetic organic chemistry concerned with making natural products. Natural products are molecules like salicylic acid that are isolated from nature. So salicylic acid, for example, is isolated from tree bark. And total synthesis refers to the fact that we're building these molecules from scratch using really simple, small molecules derived from petroleum or abundant natural sources. In undergrad, I worked on this molecule, complanidin A, which promotes the growth of neurons in a dish. And I was really interested in this application of the molecule. I was always interested in what are we going to do once the synthesis is done. And this is, in my mind, applied synthesis, an attitude toward applied natural product synthesis that I think in the past several years has really taken a firm hold in the natural product community. But I sort of, in undergrad, was struggling with what therapeutic area I would want to apply chemistry to in the future until I saw this issue of Time magazine in September 2013. Google had decided to launch a venture whose mission would be to extend the human lifespan, which completely blew my mind because I didn't think that extending the human lifespan was at all possible. And the Google's venture was launched, and it exists today. It's called Calico. And if you go on the Google Calico website, you see Cynthia Kenyon staring back at you with a smile on her face. Her lab is the lab that kicked off this whole field of extending the lifespan and manipulating aging. In the early 90s, her lab discovered that if you mutate a single gene in a roundworm called C. elegans, that you can generate a strain that lives twice as long and ages half as fast. So I read about this and about all the subsequent work that has gone on in the field in the next 2 decades, and I said, all right, this is what I want to apply chemistry to. I chose to come to Stanford to work with Noah Burns. And the reason I wanted to work with Noah is because he was excited as I was about applied synthesis, about doing interesting things with the molecules we synthesize. And we kicked back ideas back and forth about these natural products called iridoids that have also been found to extend the C. elegans lifespan. But we decided actually not to proceed with this project. And to explain why, I want to share that there are a few different criteria that we used to decide whether a synthetic target was really what we wanted to pursue. Of course, we wanted to have some interesting chemistry that we could propose, and we thought we were able to do that with these molecules. We want the molecule to have interesting properties, which we thought we had with this lifespan extension. And the last thing is scarcity. It's hard to justify synthesizing a molecule from scratch if you can obtain it in high yield from nature or elsewhere. And unfortunately, with iridoids, this is not the case. You can obtain them in high yield from plants and even in some case on the internet. So we scrapped this project, and we were unable to find other natural products with a lot of interesting data in aging or lifespan. And you can see why. These criteria that we use are often at odds with each other. If something is interesting, that means people have gotten their hands on enough of it to acquire interesting data. If something's interesting and scarce, well, it's probably already been synthesized or people are working on it. But there is one class of natural products that fulfills all three of these requirements probably better than any other natural product class I can think of, and that's membrane lipids. The membrane is the surface of every cell in your body. It's a skin that encapsulates all the cell contents, and it's made up of molecules called lipids that kind of look like this if you zoom in on them. The lipid molecule, the membrane lipid molecule, has three components. It has a hydrophilic head group that interfaces with water outside and inside the cell. And it has two hydrophobic tails, which make up the interior of the membrane. This, of course, is a cartoon. The actual structure looks like this. And the head group can vary in structure, and the tails can vary in length and in the number of double bonds in them. But overall, this is what the vast majority of membrane lipids in your body and in the bodies and in the cell membranes of bacteria look like. But the Burns lab was interested in molecules that don't look like this. For example, GDGTs, which is a class of lipids made by archaea, which are some of the most ancient species on Earth, single-celled organisms. This is obviously a very different structure with two head groups and a single tail that makes a cyclic structure. It has cyclopentanes here shown in blue. And so this was the molecule that I said, okay, this fulfills all of and Noah and I agreed, this fulfills all the requirements we have for a really exciting synthetic target. And so this is what I worked on for most of my first year in the chemistry program here at Stanford. And we listened to a lot of music in the Burns lab. And we listened to a lot of Disney music in the Burns lab. And every time Go the Distance from Hercules came on, Jaron Mercer said, this is Steven's song, because he's working on the biggest and toughest and most insane molecule in the lab. And I always appreciated that. I didn't actually end up completing a synthesis of this molecule, not even close, but Isaac Falk from the Burns lab published a really nice paper on this earlier this year. When I started this project, I thought, I really, again, still am really interested in the application of molecules. And when I finish this synthesis, I don't just want to publish the synthesis. I want to publish some interesting experiments I do with the molecule too. So as soon as I could, I started learning experiments in lipid biophysics, which is what you do to study these types of molecules. You build a membrane out of them, and then you see the properties that those membranes have. I went to the Boxer lab here at Stanford to do this, where people specialize in this kind of thing, and learned techniques from Frank Moss, who was a grad student at the time. And I didn't make it that far with GDGTs because as I was working on this, other people started completing really exciting syntheses in the Burns lab. And I was more than happy to take on that material and do interesting experiments. The first of these was this molecule, danicalipin A, another really strange structure that I started some self-assembly studies and started isolating it from algae and kicked off some experiments that ultimately culminated as a result of others' very hard work in a publication last year. But before long, another even more feasible to study lipid was completed by Jaron Mercer and Carolyn Cohen. And I should mention that Dennis Hu and Matt Landry finished this danicalipin A molecule. Jaron and Carolyn finished lateranes. And this is a really exciting structure. These ladder-shaped structures in the molecules, which is why they're called lateranes, are extremely unusual. Practically no other natural product has this. And this is made by an oceanic bacterium that has another weird thing about it, which is that it makes hydrazine. Hydrazine is a toxic chemical that would destroy any other organism. But Anammox bacteria, which make both of these things, make hydrazine in order to harvest it for energy. And we and others thought that would have something to do with it. I did some work on the three-dimensional structure of lateran lipids and the self-assembly properties that went into the 2016 synthesis paper. And then as soon as that was over, we were thinking about, can we link these two phenomena together, the lateranes and hydrazine? And we and other people in the field thought that lateranes evolved to prevent the diffusion of hydrazine, to encapsulate it and keep it from escaping the part of the cell where it's made and destroying the rest of the cell. And this is a testable hypothesis. All we would have to do is make a faked empty cell out of this lipid called a vesicle, which is just a membrane, and then wait for the hydrazine to go from outside to inside the membrane and measure the rate compared to a typical membrane like what we see here in the top right corner. Unfortunately, the technology to do this didn't exist at the time. There were some molecular sensors of hydrazine, but they were not water soluble enough to perform the assay. So I made a derivative of one of these sensors that was water soluble, and we ended up filing a patent on that, and also a patent on vesicles made out of lateranes that we were pleased to see were granted last year. And then also this molecule had suitable properties to detect the crossage or the passage diffusion of hydrazine across a membrane, and we were able to acquire the data. Surprisingly, lateranes actually do not prevent the diffusion of hydrazine across membranes, but my collaborator Frank saw that it prevents the diffusion of acid, which also has interesting implications for the biology, and we published a story on this together in 2018. But this talk is about aging, so I want to take a quick step back and just say that at the end of my second year in grad school, when we finished up this 2016 paper, I was really sort of having an identity crisis. I had become a full-time lipid biophysicist, which I enjoyed, and didn't want to go back to natural product total synthesis, but I didn't want to do lipid biophysics as a career. And if there's any time to change direction, the end of your second year in grad school is a really good time to do it. And my interest in aging had only grown and had only become more acute. In the second year of the program, I gave this second year seminar, which was a requirement for the program, on brain aging. My interest had become focused on the brain in particular. This is because of conversations with people like my grandfather and Justin Dubois. And also I had thought about how there's hope for other organs like the heart to maybe be even grown in a dish and transplanted, or prosthetic versions of the organs can be generated and put in place of an aging or failing organ. But that's not going to happen with the brain anytime soon. And so I think it's an interesting place to get in there with some chemistry. And also, scientists have done a really poor job so far of treating neurodegenerative diseases such as Alzheimer's disease that are a result of brain aging. So I really decided that it might be a time to get into this. And so I approached Tony Wyss-Coray, as he mentioned. I knocked on his door and asked if he would ever take a chemist into his lab, which he had never done before. And he said, okay. But he also said, it sounds like things are going pretty well in the Burns lab. Maybe finish up this hydrazine stuff, and that'll be a good call for the future. And he was absolutely right. I stuck around and got that paper done, and that was very fulfilling. When I joined the Wyss-Coray lab in 2017, I was able to do some synthesis, and I've continued to do some over the years, but I won't talk about that today. Some of it has resulted in a manuscript, and some of it's in progress. And I've done other side projects with mass spec and on cerebrospinal fluid with other people in the lab that, again, some have resulted in manuscripts, some are still in progress that I won't talk about. Today, what I want to talk about is my project, which is concerned specifically with brain aging and Alzheimer's disease. Our lab is concerned with why are people who are older have such a higher risk of getting Alzheimer's disease. If you plot the data on the left here in a slightly different way, you can look at the sort of two dimensions you can look at this problem through. It's very common to take samples from Alzheimer's patients and age-matched controls and analyze them and compare them to learn about the disease. Our lab does a lot of this, but we're also interested in the aging process that happens before Alzheimer's disease occurs. In this way, we're hoping to identify diagnostics that can better predict Alzheimer's disease and therapeutic targets that can prevent it. Luckily for us, a lot is already known about aging. In fact, people have broken down the different processes that occur as you age into different cellular and molecular, what we call phenotypes, phenomena. And two of them in particular are really relevant to the brain that I'll be talking about today. One is altered intercellular communication, which tends to refer to the immune system. And the other is loss of proteostasis. In order to define proteostasis, I am going to quickly define protein. A protein is just the product of a gene. A gene is a region of DNA that gets copied into an intermediate molecule called RNA, and then that RNA molecule is copied into a protein, which instead of G's, T's, A's, and C's like DNA, is made up of amino acids. It's a string of amino acids that's produced, and then it folds into some three-dimensional structure. This scheme is true, but it's also an oversimplification of the reality. In fact, a single gene can produce many different protein molecules. Mutation can result in differences in the DNA code. Splicing can result in different RNA molecules. These RNA molecules can be translated into, therefore, different proteins called isoforms. And then any of these products can undergo further chemical reactions to result in different forms of the protein, which are called proteoforms. Once a proteoform is produced, it can associate with itself or other proteins to form things called aggregates, protein-protein interactions, multi-subunit complexes, or it can associate with a small molecule or ion and form a ligand-host complex or any other sort of association. Different proteoforms and different complexes can be sort of all thought as different structural states of the protein. And these are extremely important for their biological function. There are things some of these molecules could do that the others couldn't, and important to their dysfunction. Some of them will cause disease. The regulation of the balance between these proteins, making sure there isn't too many copies of one or not enough copies of the other, is called protein homeostasis, or referred to as this portmanteau proteostasis. And as I mentioned, the decline of proteostasis is a hallmark of aging with special relevance to the brain. Now, if you wanted to look for proteoforms that were relevant to aging and Alzheimer's disease, especially in the brain, the cerebrospinal fluid would be a great place to look. Cerebrospinal fluid is the fluid separate from the blood that your brain sits in. It's a clear, colorless fluid that's biopsied by lumbar puncture or spinal tap. And it contains multiple diagnostics of Alzheimer's disease that are used clinically to classify patients, the most famous of which being amyloid beta 42. So for those who don't know, the amyloid cascade hypothesis is one of the prevailing hypotheses for how Alzheimer's disease is caused. There's a gene called APP, which makes a protein that sits on the surface of cells. Proteolysis, which means cleavage by an enzyme called a protease, makes a piece of this protein called a peptide, and in this case, amyloid beta 42 peptide. And this is the biomarker in CSF that's used to classify patients. This peptide has some healthy functions, it appears, in the brain, but sometimes it can misfold and stick to itself and form a harmful structure called an oligomer, which is toxic to neurons and that is being targeted by several drugs in development right now. And then these oligomers can further aggregate and form plaques that are visible in the brains of deceased Alzheimer's patients under a microscope. CSF tau is another protein that undergoes structural changes. Tau is phosphorylated and then fibrillizes. And I want to emphasize that these proteins are undergoing structural changes and that they happen as you age. And these changes are super relevant to the disease pathogenesis rather than some gene expression value. There are other examples in the CSF. There are more proteolytic cleavage products. There are small molecule binders of CSF proteins that are known to be elevated in the CSF of Alzheimer's patients. And what hasn't been done before is taking a step back and looking at any of these types of structural changes and saying, what's changing as you age? So this is the goal of my project, is to do an unbiased screen and look and see if we could find anything new in an unbiased screen. I've been talking about structure, but actually even looking at simple protein abundances, which is more conventional in studying proteins, hadn't been done with aging in the CSF of mice. And in fact, the studies in humans had been extremely limited as well. This was alarming to us because a lot of what we understand about how the brain ages and how neurodegeneration develops, and then how we can actually rejuvenate the brain, has been done in mice. In particular, our lab has found that if you expose an old mouse to blood proteins from young mice, it rejuvenates the brain. And this is very exciting and surprising because the blood is separated from the brain by the blood-brain barrier. We thought that the CSF, the CSF fluid, the CSF, which is in direct contact with the brain, likely has proteins that are even more relevant to aging and Alzheimer's disease in the brain. So we wanted to look at these protein abundances. And we also, of course, wanted to look for structural states. And this, of course, has never been done in CSF with aging, but actually looking at changing structural states across all proteins that you can observe hasn't been done in any organism, in any tissue with aging. And so we were excited to explore that. Part of the reason it hasn't been done is because high throughput structural biology is hard. And there wasn't any technology to do this kind of thing until 2014, when Paolo Piccati's lab came out with a method called limited proteolysis mass spectrometry, or LIP-MS. Before I explain how LIP-MS works, I'll just show that it can detect changes in structure, like aggregation or in conformation, going here from a helix to a loop. It can detect changes in many proteins that are not revealed by abundance. So here in this plot, you have proteins that are changing in abundance that are outlined by red or green. And then you have boxes filled in by blue that are undergoing structural change. And there are many blue boxes that don't have outlines. So we're getting information with this method that we wouldn't get from a normal protein abundance experiment. And then using the 3D dimensional information that we already have about proteins, we can take LIP-MS results and look where on the protein structures are changing. So here we see signals of change adjacent to small molecule binding sites, suggesting changing in binding. So in order to explain how LIP-MS works, I have to explain mass spectrometry-based proteomics, or mass spec. The proteome is the set of all proteins in whatever you're looking at. In my case, it's CSF. Proteomics is the study of these. Mass is exactly what it sounds like, the weight of the molecule. And spectrometry refers to the fact that you're measuring spectra, which are these graphs in the bottom right corner of the screen. The x-axis here is m over z, where m is the mass and z is the electrical charge of what you're seeing. And the y-axis is the intensity, which correlates to the copy number of the peptide. So this is different for every ion you're looking at. You can't compare these different peptides to each other. But in this cartoon, I've shown that the peak for the red peptide is lower than the peak for the other peptides because there are more copies of the others. When you do a mass spec proteomics experiment, you take the proteome, in my case CSF, and unfortunately we can't shoot all these onto the mass spec at the same time because they're too different from each other. So first we have to treat them with a protease, a protein cutting enzyme that generates peptides, which are pieces of the protein. And when we inject it, two different things happen. One is that the mass spectrometer measures the intensities of the whole peptides. And then the other thing is that peptide by peptide, it selects ions to fragment and then does mass spectrometry on the fragments. And those fragments tell us what the sequence of the peptide is, what amino acids are in the peptide and in what order. The peptide sequences tell us what protein the peptide came from. And then the peptide intensities can tell us about the protein abundances in your sample. And this is really what we're getting at. If your brain is making more or less of a protein as you age, that's interesting to us. In case you're curious, here's what the hardware looks like. There are just 3 pieces. The LC has an autosampler that picks up the peptides and then it pushes it through the column. The column separates the peptides and sprays them into the mass spec. And this is very important so that we're analyzing a different peptide at different times. We might analyze a single sample over 2 hours and we're seeing different things throughout those 2 hours. And then the mass spec does the analysis. On the sample prep side, when you take your samples, an experiment might look like this. So let's say I have samples of CSF from young mice and CSF from old mice, and a protein changes in abundance. This is just abundance. This is the most classic type of mass spec proteomics experiment. The first thing we do is unfold the proteins with detergent and heat. This is called denaturation. And then we use another protease, we use a protease called trypsin that cleaves these into peptides. And what trypsin does is it cuts these proteins after arginine, which is represented by an R, or lysine, which is represented by a K. And what this generates is molecules we call tryptic peptides, or fully tryptic peptides, where both cleavage sites were after an R or K. If your trypsinization is performed properly, you can assume that the copy number of the protein is equal to the copy number of the peptide, as shown in this cartoon, which means when you take the mass spec intensities of the old versus young peptides, for example, you can get information about the relative protein abundances as well. With LIP-MS, let's say that in addition to abundance changes, you have a protein that doesn't change in abundance, but it changes in structure. So in this example, you would have a small molecule not bound in young CSF and a small molecule bound in old CSF. We detect this change using LIP-MS by treating these proteins before we unfold them with yet another protease called proteinase K or PK. And in this example, the binding of the molecule might protect the protein from PK cleavage so that it doesn't cut the bound structure and it does cut the unbound structure. This means that after the treatment with the enzyme, we might see a cut in our protein down here and no cut up here. PK, because unlike trypsin, it is not an amino acid selective protease, can just cut anywhere. And so in this example, you might see a cut after an A, which is not an R or a K. And then we do the rest of the sample prep like normal, using doing a denaturation and cleaving after R or K with trypsin. The result is peptides called semi-tryptic peptides, where one of the sites is tryptic. So in this example on the left, you have an R or K before, but not an R or K at the end. And this peptide on the right-hand side has an R or K at the end, but not an R or K before. These semi-tryptic peptides are evidence of decreased protection from PK, and fully tryptic peptides are evidence of increased protection from PK, and that's how we get structural information out of the experiment. The whole experiment looks like this, and we end up with 4 sample groups that we're comparing, 2 from your test group, in my case, CSF from old mice, and 2 from the control group, in my case, the CSF of young mice, each with PK not treated and one with PK treated. So I denote this PK minus for just the good old abundance changes, and then PK plus for the samples that were looking for structural change. I had the incredible and distinct privilege of being sent to the Picotti lab to learn how to do this technique. Thanks a million to Tony for supporting me in this. And I learned from Marie-Therese Macmul, who's a postdoc there, how they do the sample prep and how they do their data analysis. I brought those techniques back. I collected cerebrospinal fluid from young and old mice. Huge shout out to Tali Ram from my lab, who developed the stereotactic version of this CSF collection that works really well. And after generating the samples, I split them into one duplicate to treat with PK and one not. And then I did the rest of the denaturation and sample prep as normal. And then I had these samples analyzed in 2 different acquisition modes, which I won't describe the difference between these two, except to say that DIA is just a better method. It generates higher quality data with a little more effort. Very sadly, even though I'd been running samples from others in the lab all fall, the instrument went down. It broke down for 2 whole months, and then we had to move the instrument to the new building, and then the pandemic hit. So I was not able to run these samples myself. So 1,000 thanks to Ryan Lieb and everyone at SUMS for acquiring the raw data for us. And then I took the raw data and did the analysis as is typically done. And I got a surprising result. Even though the DIA data, as predictable, had higher quality metrics, it identified more lit peptides and identified peptides in more samples, and the quantitative precision was comparable. We got way more hits with the DDA data, and I looked into every different way we could look at this data to see if there was any problem with the quality of the DIA data, and could find nothing. And so when you have a data set with inferior quality but more statistically significant results, you start to wonder, could these be false positives? And I started looking in all the differences between these acquisition modes. And the source of these differences, I think, were the different softwares that you use to analyze the data. And overall, these softwares work very similarly, except for one part, which is the protein grouping algorithm. And we landed on this as a major source for this disparity. So what is a protein grouping algorithm? Well, I've given some simple examples today of a single protein is cleaved into a single peptide and it matches and you're all happy. But it's not uncommon at all to have peptides that once you find their sequence, they actually match different proteins. So this is an example where you have proteins and up on top and peptides on the bottom, and these edges connect where there's a sequence match. So the question you would ask in a situation like this is, which proteins were actually in the sample? You could say, oh, I have 6 proteins that match, so I identified 6 proteins in this cerebrospinal fluid. But the field would say, no, actually, you didn't necessarily detect proteins 2 and 4 here. You don't need these proteins to have existed to explain the 7 peptides you got. And so protein grouping deals with this, and different softwares deal with it differently. But what they all do is they generate protein groups, or PGs. And they're called protein groups because multiple proteins can be in them. In this example, 5 and 6 are in the same protein group because they have the exact same level of evidence for their existence. You can't pick one over the other. Why is this important? It's important because sometimes in LipMS, you have a protein that changes in structure and abundance, and you have to correct for that, which involves the protein groups. More specifically, let's say you have a protein that doubles in abundance with age. If your PG is a good approximation of that protein, then its intensity on mass spec would go from a relative number from 1 to 2. If you have a lit peptide that increases to a lesser degree, then that would actually mean, for example, that you have decreased protection from PK, but you wouldn't see that in your spreadsheet. You would see a positive, or you would see a number above 1. You would see 1.5. So you have to divide by the protein group intensity, and that would give you the number that you're actually looking for, something less than 1, to show you there was decreased protection. The thing I want to emphasize here is that what you're looking at, this ratio, this normalized lit peptide intensity, is what we do all our statistical tests on, and it's what we extract our results from. And the numerator is just one peptide. That one peptide tells you everything you want to know about the structure of your protein. The denominator is calculated from multiple different proteins, or peptides rather, and they're combined into the PG. And so we started to ask, do the MaxQuant and ID-Picker PGs do a good job of this? Do they remove the protein abundance well? And one way we decided to look at this is by looking at the PK minus peptides. So here we have not added PK, we're just doing a regular old protein abundance experiment. And every peptide abundance should be similar to the protein abundance. So if your protein group doubles with age, you should expect your peptide to also double with age. And so dividing all of these should give you ratios close to 1. And you wouldn't see any statistically significant changes. These are called volcano plots, where the x-axis is the logarithm of the fold change, where the fold change is the ratio of old to young intensity. So if you have a change of 1, the logarithm would be 0. And as you get farther away from 0 with stronger changes, you get higher on the y-axis, which is statistical significance. So greater changes mean greater statistical significance. What we found is that when we divide PK minus peptides, this is in the DDA data set, by all the protein groups, there are statistically significant results, even though there shouldn't be any in an ideal world. And sure enough, when you look at the PK plus data, where now you're trying to look at structural changes and you're excited to see, did any of my proteins change in structure? Well, sure, we have some peptides that have statistically significant difference in age, but you also got the same result without even adding PK. So you can't be sure whether this is a structural change or not. And one way that this could happen is highlighted by this protein that this protein group that I've circled in red. These 3 CHGB peptides out to the left are neutralized effectively by the protein group division method. But there are several other peptides in the protein group that are sort of artificially pushed up into significance. So the problem in this particular case is that this PG is a large and diverse peptide group. The smaller and more similar peptide groups are, the less of a problem this will be. So when we look back at how the protein grouping algorithms actually work, we realize that there could be situations where your numerator peptide and your denominator peptides might not come from the same protein. So in this example, for example, in PG1 in the MaxQuant case, you could have your numerator be peptide A, which can only come from protein 1. But peptide B, which could have come from a mixture of proteins 1 and 2, could be offset, and the abundance of protein 2 could be affecting the accuracy of that peptide, or at least it's not approximating the same thing that A came from. ID-Picker does the same thing too, a little differently. And I want to emphasize here that there's no problem with using these protein groups for protein abundance. In particular, I'm going to present some protein abundance data later that uses ID-Picker protein groups, because these do actually a really good job of approximating real proteins. The problem here is that since we're looking at a numerator peptide that might be weird, it might come from a different protein that isn't really at the center of what a typical protein group would look like, we thought we could design our protein groups with LibMS to look a little more similar to the numerator. And the simplest, stupidest way to do this is to just make sure that all the peptides in the protein group map to the exact same set of proteins. So in this case, A would get its own PG because it's the only peptide that only matches protein 1. B would also get its own PG because it's the only peptide that matches 1 and 2. C and D would be in the same PG because they're the only peptides that match 3, and so forth. And what we get when we do this is smaller and less diverse protein groups, which is the exact opposite of the large and diverse protein groups we were looking at before. These histograms up top show that there are fewer peptides per protein group, and the histograms at the bottom, I've plotted along the x-axis this standard deviation, the spread of log fold changes per protein group, and that's also reduced. So things are more similar to each other. Encouragingly, when we use this method of dividing proteins as opposed to ID pickers, we also have increased power to detect changes in our data set, suggesting that this better matching is also advantageous for detecting changes. So I implemented all of this into a script that you can put all of your analytical parameters in at the top, and it runs everything from beginning to end, and it generates a statistical score for each PG of structural change. Because you need to know the R programming language to use that script, and because it's not easy to save and load and keep track of your parameters, I also made a desktop app for Windows that allows you to save and load your changes and point and click and run without knowing how to program. And I actually found this very useful for my research so that I could easily do systematic comparisons of my analytical parameters. And when you make an app, you have to name it something. So I named it the LipMS Test Control Screening Platform, or LipTex. So the idea here is if you really have any data set where you have a test and control group with PK and PK minus and PK plus conditions, you can plug it into this software and it'll run. When I ran this analysis on my data, I identified first abundance changes, and then I'll talk about structure later. But I identified 38 protein groups that change in abundance with age. Most of these relate to the immune system or to the extracellular matrix, and I wish I could talk about them all and explain the potential biological implications of each one of them. But I'll just stop to show that any of these could be a potential candidate for a biomarker of aging or something that you could try to manipulate to rejuvenate the brain. What you can see is that this whole plot is dominated by proteins that start with Ig. This means immunoglobulin, and it just means antibody, just like the antibodies that COVID-19 stimulates. And the antibody that increased the most, or rather, this is technically a piece of an antibody, is IgHM, which is a piece of a type of antibody called IgM, which is one of the 5 types. And this was surprising to us because IgM is typically what we call a naive B cell state, where when you encounter an antigen for the first time, you might produce IgM, but then as you encounter it more and more as you age, then you would expect those B cells to switch to a different type, from IgM to IgG. So the fact that this is increasing with age is interesting, and IgM is often associated with autoimmune disease and multiple sclerosis. So there might be something autoimmune happening in the brain with aging. Looking at these individual statistical scores next to each other, we thought, well, are any of these changes correlated with each other? Are there any PGs that seem to change with aging in a similar way? To look at this, I did an analysis called Weighted Gene Co-Expression Network Analysis, or WGCNA. And here I've generated a heat map where every pixel is brighter if the 2 proteins are more highly correlated. And I'll just point out a couple of things. One is that we saw that proteases, these are enzymes that cleave proteins as a part of their natural function in the CSF, increased with aging in a highly correlated way. So they might have been produced by the same cell type, perhaps microglia in the brain. And inhibitors like cystatin-3 and serpin-C1 were also changing with age, and cystatin-3 was highly correlated with cathepsin D. So there might be sort of a co-regulatory change here happening where proteases are increasing with age, but also the inhibitors of them are. And maybe some of these proteases are successfully inhibited and some are not. I also observed a high correlation between IgHM, which is a piece of an IgM antibody, and another antibody piece, a variable chain, 126, that you can't tell which type of antibody it came from. But the fact that it's so highly correlated with IgHM suggests that these might be part of the same IgM molecule that is increasing with age. And so if this is one, what we call clone of this antibody that's changing with age, it raises further questions about what antigen it could possibly be reacting to. Turning from abundance changes to structural changes, I got 16 statistically significant changes with age using the LipTex software. And I filtered out these hits using the PK minus peptides as I described before. I wanted to make sure that all the PK minus peptides had intensities that were clustered nicely around the PG intensity. And I also wanted to make sure that there was nothing significantly changing after doing that division. So by doing this, I identified 6 proteins that we were really highly confident in. And in LipMS, usually what you do is you now take the existing information that we have about the 3D structures of these molecules and map on the structures that we have observed and learn something about the structure. The 3D structures of kininogen 1 and fibulin 1 are not known. Fibulin 1 corroborates the changes that we're seeing in the extracellular matrix, whereas kininogen 1 has a known cleavage product that is known to increase in abundance in CSF of Alzheimer's patients. And this type of increase in cleavage would also explain the decrease in PK protection we see in mouse aging. So this is a potential novel link between mouse aging and human Alzheimer's disease. 3 of these 6 hits had the same thing, potential links to human Alzheimer's disease. We observed increased protection at a hyaluronin binding site on ITIH2. And hyaluronin is a molecule known to increase in abundance with CSF. So this could be coming from increased abundance in aging, which is another link between mouse aging and human Alzheimer's disease. LPPLA2 is an enzyme that makes LPC. It cleaves full PC molecules into LPC. And the increased protection site we saw is sandwiched between the lipid binding site and the catalytic site, suggesting that this could be binding of any lipid, but LPC is known to increase in the CSF of Alzheimer's patients. And so if there's product inhibition by this molecule in mouse CSF, that would be another link. And then lastly, 14-3-3 proteins are known to participate in kinase signaling by binding to various phosphoproteins. And we observed increased protection right next to a phosphopeptide binding site. Protein phosphorylation is also known to increase with Alzheimer's disease in the brain. And so this could be another link between mouse aging and human Alzheimer's disease, with 4 total. Lastly, I want to talk about the top hit that came out of the screen. It's a molecule called CD5L. It's a gene made by macrophages, which are the cells that devour pathogens in your blood. When a tissue is infected, macrophages have to exit the bloodstream and go into the tissue. And this is one of the proteins that they secrete in order to help each other survive in that environment. But separate from that phenomenon, the CD5L molecule is known to circulate in the blood, not as a free molecule, but as a covalent complex with IgM. And this complex is known to have bioactivities that are different from free CD5L and different from the IgM molecule itself. In particular, in obesity, it's been found to stimulate production of autoantibodies. This is, again, auto meaning that the immune system is attacking the healthy body. So this has a negative effect in obesity. The CD5L molecule, which is also called AIM, the ratio of abundance between that and the complex has also been used as a biomarker for liver cancer, suggesting again that these have different biological activities. So we wanted to know if this structural information had anything to do with our lip results. And the complex was actually not known to exist in the cerebrospinal fluid. We performed, well, I performed a Western blot. A Western blot separates objects by size. So the y-axis here is size. Proteins that are, or objects that are of a larger size will appear higher on the blot. And then on the x-axis here, each row of the first 8 is a different sample that I collected either from young mouse CSF or old mouse CSF. I also ran a free CD5L molecule, which runs down here because it's relatively small. If you add IgM to one of those molecules, it generates a small amount of this complex. And on Western blot, you only visualize one type of molecule. So we're only seeing CD5L molecules here. The IgM molecule itself doesn't show up on the blot. So as you can see, we see that the complex does exist in cerebrospinal fluid. And more than that, it increases significantly in abundance with age, which is very interesting to us and gratifying because this is what we were looking for at the beginning of the study, more advanced gene products like complexes that change with Alzheimer's or change with aging. And we would not have made this discovery without LipMS because CD5L wasn't one of our abundance hits. So to summarize this, I utilized PK minus control data to take a bit of a deeper look at LipMS analysis. The simple fact of dividing PK minus peptides by their protein groups hasn't been done before in the LipMS literature. I generated software for analyzing LipMS data, which is also missing from the LipMS literature. And I used it to measure protein abundance that change in the CSF with aging. And I found 6 high confidence hits for changing structural state with age, including 4 potential new links between mouse aging and human Alzheimer's disease. I found a new age-associated protein complex in the CSF, CD5L IgM, pointing to potential relevance of B cells in mouse brain aging and a potential autoimmune role. And with that, I will take any questions from the audience. Yeah, no, Justin, we have a mic for people to ask in person so that people in the room, or on the Zoom rather, can hear you. So thanks, Tony.
Move it around on this microphone. Stephen, that was a fantastic talk, and you've come a long way from complanidine. My question is a little bit, off the topic of your talk, but can you transfuse CSF between young and old mice, and what happens relative to transfusion, blood transfusion? Yeah, it's a great and relevant question, and you can stay tuned because my lab mate Tali Ram has done exactly that. I mean, she's, maybe I shouldn't be spending so many words on this in my defense, but she's done an incredible job of surgically implanting pumps that can do exactly this. I mean, she went through so many iterations of that and saw some really interesting results in a particular cell type. I won't give anything away, but yeah, there are definitely changes that look beneficial. What are you defending? She's a postdoc, unfortunately. I'll send you the paper when it comes out. I'll be a co-author. I'll take it. Yeah, sure. Actually, Tam, can you take it? Any questions from Zoom? Oh, Oliver's got a question.
Thanks, Stephen, for the beautiful talk. Is it on?
It's on.
It is. Cool. So the CSF is a little bit like, a juice that flows through the brain in very simple terms. So it gets a lot of input from both the periphery and also from the brain itself. It makes sense to sample it for humans, but I mean, you could argue that when you sample it from your mouse, the mouse doesn't just walk afterwards, right? Yeah. It's pretty much done. So wouldn't it be sensible, or also as a next step, to actually do this kind of protein conformational screening actually in the parenchyma itself, actually maybe even with purified cells? If you think about Alzheimer's, there's a huge debate about how protein compactization and so on happens within the cell or in a specific cell type, maybe a type of neurons first, and then starts to accumulate and spread and whatever. Is that maybe something that could be interesting to look at, and what would keep you from doing that? Yeah, I've laid awake at night wishing to do that experiment. So Oliver's talking about not looking at the fluid, looking at the brain, because that's where these protein aggregates are, in the brain tissue. So people in the Piccati lab are working on doing that. It's a tough thing to do with an enzyme, with PK, but for a lot of reasons. But I actually, as one of my graduation requirements, presented a proposal to do exactly that, not using an enzyme, but using a reactive small molecule that, instead of creating a cut, would create a modification on the proteins that you could detect. And the rates of that reaction would almost certainly be structure dependent. It's a very exciting possibility, but I think with LipMS, it's not there yet. You don't want to follow up right away with the second one. Do you think there's some sort of an overall direction, whether the protein itself, the proteome, would get more compacted? Would you see it makes any difference? Which would be more or less accessible over time, that the proteins get more denser or whatever? Or is this just going to be always some up, so down, or do you think there could be an overall trend over time? Have you seen anything in your data? Yeah, it's a good question. I mean, I didn't detect enough changes to see that type of trend. So as you can see, I saw 4 that increase in protection, which is exactly what you're talking about, more shielded to cleavage, and 2
that decrease. And you know, if you did a you know, a binomial test to get a p-value of this, it would not be 0.05. So I don't know, but it would be interesting to look not only at that, but you know, just spin down proteins in the CSF and see if there are larger complexes forming. Yeah, that would be really interesting. Okay, I think I should move on then to the acknowledgements. Yeah, any questions from Zoom?
Oh yeah, I'll ask one, Stephen.
Thanks, Peyton.
I have a question about how the IgM gets there. Like, is the fact that there's more IgM a byproduct of an increase in the whatever is involved in its transport, or is it, you know, what is the aging related impact of how the IgM has gotten there?
I love that question. So I don't know. I don't think that IgM, it's controversial whether IgGs can cross the blood-brain barrier. And IgM, as you know, is 5 times as big. So it's not my intuition that the IgM is crossing the blood-brain barrier and getting into the CSF. Although other data from our lab from mouse plasma suggest that IgM does change in the plasma as well in mice. What I think is that there are B cells sitting in the meninges that are producing this. And a really exciting report just came out earlier this year on B cells that live in the meninges and where they come from and how they get educated by CNS antigens. And that's where my money would be.
Thanks.
I actually have one question that's kind of a follow-up to Peyton's. This is Emma. Hey, that was a great presentation, Stephen. You spoke a lot about how some of the changes that you've seen structurally seem to result from changes in cleavage. And I'm curious if you have looked at all if any of the changes that are occurring might have any commonalities in their signal peptides and how they're actually secreted, because I'm curious as to whether or not signal peptides would actually show up in your mass spec. And if not, if that could actually be sort of informing hypotheses about whether or not the secretome itself is being changed, and whether or not that's something you can see with the data that you have.
Yeah, that's a great question, Emma. I've never, I haven't looked at every peptide and every PG I've analyzed, and I've never seen a signal peptide come out of mass spec before. And there might be reasons for that there might not be a lot of Rs or Ks in signal peptides. I'd be purely speculating, but it's a really fascinating idea. I mean, it wouldn't be too hard to look at simple sequence differences in things in terms of their signal peptides. I don't know if the sequence itself affects secretion that much, but it's a very interesting idea.
Awesome, thank you.
Thanks. All right. Oh my gosh, I have so many people to acknowledge because I did not do any of this alone. It would have been completely impossible. And the first people I want to thank are my family, especially my parents. I mean, mom and dad, I couldn't have done any of this without you even close. I'm the luckiest person in the world to have had you as parents, and there's zero chance without either of you that I would be here. So thank you for everything. Very fortunate. Scott, thank you so much for always believing me and never doubting me, and giving me great advice in life, and also giving me the best recommendations of art and entertainment to enjoy in my free time, and completely blowing my mind on the podcast. Sarah, I really do not know what I would have done without you these past 7 years. Having you in my life has been such an important part of my support network, and I've just gone through things that I don't know what I would have done without you, so thank you so much. Wish Grandpa Jack could be here. He sadly passed away in 2017, and he was such a huge fan of my science, and I miss him so much, but I think he would have had a good time today. To my grandparents and aunt and uncles and Eliza and the kids and all the cousins, it's impossible to state how important it is for me to be able to come back on vacation and have you guys support me and say you're proud of me and say that everything's all right. It's a huge part of why I do what I do, is to try to make you guys proud, and that can't really be overstated. I want to thank my friends from high school and college who have kept with me all these years and kept supporting me. I'm amazed to see that Nick Arthofer joined us from Prague. I really want to thank John Gerlach and his family for hosting me in Berkeley and Healdsburg over the years. The best New Year's Eve parties I've ever had. And Ben Sugar's family for giving me another second home in Monterey and taking me on an Alaskan cruise, and all the love and support, and especially Dr. Shug for trying to convince me to be a medical doctor after grad school, which I definitely won't do, sorry. I just have to thank people in undergrad. Most of them are not here, but I mean, this is how I became a scientist, is by following the lead of people like Richmond Sarpong and Sarah Reisman and everyone in those labs. Special shout out to Lisa, who I met in my summer at Caltech, who helped me with protein modeling that you saw in this talk. So thanks, Lisa. And thanks to Teresa and Matt for being yet another place of domestic bliss that I could escape grad school to, and for having Elijah, who's my favorite little buddy. The boys, I mean, what can I say? Living with you guys on Comstock Circle for 7 years, or for 6 years, and then having you on Halo for the last year has been really, I mean, like, you know, I'm the luckiest person in the world multiple times over, and you guys are a huge part of that. I mean, it was just pure joy living with you, and thank you for everything. And thanks for Katie and Adam and Natalie for being there for me always, and especially during the pandemic. And thanks to my friends in the psychology department, the Patchy Pals, which we are called for pizza-related reasons. I recommend everyone in the audience to make friends with psychology people, because they're very nice. Thanks to my neighbors in EV for giving me a sense of community, and for all my other friends at Stanford for being a constant source of support and good memories. To everyone in the Burns lab, I mean, you know, I didn't talk about it that much in my defense, but my years there were just as important to my PhD as any of the other years. And Noah, you know, we had such a good time working together, and I just have unforgettable memories there, and it was a great experience. And to Justin for knowing me since undergrad and supporting me all this time, and for helping me with advice and sounding me out, or having me you know, bounce things off him when I was considering switching labs and all that. Oh my gosh, the Wyss-Coray lab is a great place to do research. It's a lovely, lovely place. Everyone is so kind and welcoming and friendly and helpful and intelligent. And I have to specifically thank Tal again for her work on CSF, and Sophia for helping me run the mass spec for a while, and she'll be doing a bit more of that in the future. Umara and Benoit and Oliver for helping me figure out where these proteins might be coming from. Christina for conversations about lip-MS data analysis, and everyone else for just being a constant source of inspiration and humor and friendship throughout the years. It's really a lovely place. And I found this paper of the same, I found this video on my phone of the same lab retreat that I just had so much fun there. And these people who are changing are long gone, but they're much better than I was at putting on the gowns and stuff. I have to thank all my collaborators from my work from my years in the Wyss-Coray lab who helped me out. So Josh and Nicholas, I learned mass spec from you, and thank you so much. Daniel Itzhak taught me how to do chromatography like a pro. Again, have to thank Paula and Marie for hosting me and mentoring me in Zurich. Thanks to everyone at SUMS again for being great collaborators, for Anne for chairing the committee, and also Anne's lab has just done amazing work in genetics, really 21st century iterations of the Cynthia Kenyon stuff I talked about before. Her lab is doing just amazing work in lifespan and genetics. And Carolyn Bertozzi, I am such a huge fan of yours, Carolyn. You're an absolute hero to me, and I really am grateful that you exist, so I have someone to look at and say, I hope my career is something like that someday. I really look up to you, and thanks again, just like Justin, for helping me with the lab switch. And everyone in their thesis defense, thanks Steve Lynch for help with the NMR, and I'm going to do it too. He's amazing and deserves every one of these acknowledgments he gets. We couldn't do NMR without you. And I totally forgot to put Roger on here, but Roger, my goodness, you're a hero as well. I'll just give this again next year and put you on this slide. Last, but of course not least, I have to thank my incredible girlfriend, Tamara. The past year and a half could have been terrible. It could have been just awful and way worse than it was. I mean, it was very hard, but because you and I were together, we just had nonstop fun and joy, and you're so supportive, and I mean, yeah, you made all of this like way easier than it could have been. And I can never, ever, ever thank you enough. And thanks to the Chans for being another home away from home, and for buying Chloe Korgdashian their adorable little tricolor corgi, who's one of my best friends. I love you, Tam, and I'm really looking forward to facing the next phase of life with you. And thank you all for your time and attention. And for those who are in the area, we'll be meeting in Pierce Park at around 4 o'clock. So I think now we have to kick everyone out of the room, and Carolyn will stay with us for the private portion on Zoom. And if everyone else could log off of Zoom, that would be great.